started discussing a few things with regards to the acquired immunity or the adaptive immunity, right? So we saw a few basic things pertaining to acquired or the adaptive immunity, right? So just a quick look back of what we have seen in the previous session, and then we'll continue with today's topic, right? So we saw that acquired immunity is nothing but the immunity which is developed in an individual during the lifetime of that particular individual. We started calling it as acquired immunity, right? So just like, unlike the inborn immunity, what we saw, the innate immunity, which is seen in an individual by birth, this sort of immunity, that is acquired immunity, is not seen in an individual by birth, but it is usually seen in an individual during the development or during the lifetime of that particular person. Therefore, we start calling it as acquired immunity. So the person acquires this sort of immunity whenever they are exposed to some sort of a pathogenic substances or any foreign particle that has entered into our body. Right. So the immunity that is developed in the body in response to any pathogen or any foreign particle is what we call it as acquired immunity, or we also call it as adaptive immunity. Right. So we saw how exactly this sort of immunity work or this sort of immune system usually works, right? So it usually implements something called as the third line of defense, right? So we saw about the first and the second line of defense when we were talking about innate immunity. So when the first and second line of defense fails, the third line of defense, right, which is actually a part of the acquired immunity, will start operating, right? So what does this third line of defense do? It first recognizes the pathogen, right? Then it attacks the pathogen, then destroys the pathogen, and it starts remembering the pathogen that has entered into the body. That's how the third line of defense or the acquired immune system usually does. Right. Now, how do they act against a particular antigen? They start producing or synthesizing certain specific cells or specialized cells as well as antibodies. So these specialized cells as well as the antibodies would bring about the destruction of any pathogen or the foreign particle that has entered into the body. We also saw that this sort of immunity usually it takes a long period of time. So why does it take time? Because it has to recognize the pathogen, then it has to synthesize specific antibody against a specific pathogen that has entered into the body. So we started calling it as what? Specific immunity, because of its specific function in synthesizing a particular antibody, and also it usually takes a longer period of time compared to that of innate immunity. Right? Then along with that, we saw a few properties or unique features of the acquired immune system. 4 unique features of the acquired immune system. One was specificity. It is said to be highly specific in recognizing a particular antigen or pathogen that has entered into the body. Second one, diversity we saw, right? So it has the ability to recognize a diverse type of pathogens or antigens that has entered into the body. Third one, discrimination between self and non-self. So this sort of immune system is able to recognize between the self cells as well as the non-self cells. Therefore, they go and act against the non-self cell or the foreign cells that has entered into the body. And the last one, what we saw, one of the most important characteristic feature or peculiar feature of this particular acquired immunity is memory, right? So whenever there is any antigen entering into the body for the first time, this normal process of destruction usually occurs. After that, when there is a second time the same antigen enters into the body, this time the immune response is said to be much, much faster due to the activity of the memory cells, which usually recognize the antigen that has entered into the body. So this again, after this, we started classifying this immune system into 2 different types: primary immune response and the secondary immune response. Primary immune response, we said that first time whenever an antigen enters into the body, the response that is evoked by the acquired immune system against that antigen or pathogen, we started calling it as primary immune response. Secondary immune response, second time whenever the same pathogen enters into the body, at that point of time, what would happen? This time, rather than synthesizing and activation of this antibodies as well as the specialized cells, the memory cells will directly start targeting that pathogen that has entered into the body. So we also said that primary response is usually it takes longer time. Second one, it is said to be feeble and it declines fast. Whereas the secondary immune response, we also call it as anamnestic immune response. And this sort of immune response, we said that it is usually faster, right? It is a heightened immune response and it usually stays for a longer period of time. right? So that is what we have actually seen in the previous session, right? So in today's session, let us take up with the cells of the acquired immune system, right? So what are the constituents of the acquired immune system, or rather what are the cells of the acquired immune system? Similar to the barriers what we saw, right? Barriers of the innate immunity. So those were the components of the innate immunity, the primary, the first line of defense and second line of defense. So today what we are going to take up in this session is the constituents of the third line of defense, or rather we can put it as the cells of the acquired immune system, right? So let us take up with the cells of the acquired immune system. Right. So talking about the cells of the acquired, immune system. So basically the type of cells that are actually a part of acquired immune system are basically lymphocytes, right? So lymphocytes are the basic cells of the acquired immune system, right? So lymphocytes are the major constituents of the acquired immune system, right? So we have two different types of lymphocytes being produced in our body, right? So we have basically two different types of lymphocytes being produced in our body. One, we call it as B lymphocytes, and the other one is called T lymphocytes, or simply we can call them as T cells also, right? So they are either called as B lymphocytes or B cells, or we have T lymphocytes or T cells, right? So these are the two basic types of cells which are actually a part of the acquired immune system, right? So the most of the major constituents of the cells of the acquired immune system is this lymphocytes. Two major types of lymphocytes in our body, B lymphocytes, and the other one is the T lymphocyte. Now, these two types of lymphocytes, that is the B lymphocyte as well as T lymphocytes, they are being formed from something called as hematopoietic stem cells, right? Hematopoietic stem cells, right? So these cells, that is the T and the B lymphocytes, they are formed from certain specialized type of stem cells which are called as hematopoietic stem cells. So most of the cases, both of these cells, that is T and B lymphocytes, they are usually formed in the bone marrow, right, from the hematopoietic stem cells. So again, when we talk about bone marrow, we know that there are two types of bone marrows being present. One is called the yellow bone marrow and the other one is the red bone marrow, right? So yellow bone marrow is usually present in the shaft region, whereas towards the either ends, the epiphysis part, we have the red bone marrow being present. So in the red bone marrow, from these hematopoietic stem cells, we have both the B lymphocytes as well as the T lymphocytes which are actually being formed, right? So major constituents of the acquired immune system is this lymphocytes. Two types of lymphocytes, the B lymphocytes and the T lymphocytes. Both of these lymphocytes are formed from the hematopoietic stem cells, which is present in the bone marrow, more precisely speaking, present in the red bone marrow. Right. Now let's get into the details of these two lymphocytes. So we'll see what are these lymphocytes, what do they do, what do they produce, and how do they function in bringing about the destruction of the pathogens that have entered into the body. Right. So let's first begin. Right. Let's first begin with the B lymphocytes. So the first type of lymphocytes, what we'll be talking about is the B lymphocytes. Right. So B lymphocytes. So B lymphocytes, or we can either call them as B cells. Simply we can call them as B cells or B lymphocytes. Now, B lymphocytes are actually produced in the B cell, in the bone marrow, right? So they are produced in the bone marrow. So when we say bone marrow, be very clear, it is the red bone marrow and not the yellow bone marrow, right? So B lymphocytes are produced in the bone marrow. Second thing, they are matured in the bone marrow itself. right? So the process of formation as well as maturation of the B lymphocytes usually occurs in the bone marrow. That is the red bone marrow, right? So the process of production of B lymphocyte as well as maturation of B lymphocytes both occurs in the bone marrow. From which cells they are being formed? They are formed from the hematopoietic stem cells that are present in the red bone marrow. Right. Now, the term B lymphocytes, many a times we get confused telling that B lymphocytes, why the name B has been given, it is because of the bone marrow. But that is actually a false statement, right? The term B is actually derived from something called as bursa of Fabricius. Right? Bursa of Fabricius. I hope all remember this term called bursa of Fabricius. Right? If you could recollect first year when we were talking about animal kingdom, under that we spoke about one class under vertebrata, right, which was called as aves, birds. Right? So in the birds, there is one specialized organ called bursa of Fabricius. Right? First, these lymphocytes called B lymphocytes were first recognized or first identified in this particular organ, that is bursa of Fabricius in birds. That's the reason why they have taken the first letter of this and called it as B lymphocyte. So don't get confused. B does not stand for bone marrow, but B usually stands for bursa of Fabricius in case of birds. From there, then they identified that even in case of humans also, right? Even in case of humans also, in the bone marrow, we have these sort of lymphocytes which are being produced, which is called as the B lymphocytes. Right. Now, what is the role of this particular B lymphocytes in our body? So, what does the B lymphocytes do? So, let us take a few things with regards to the B lymphocytes. Let us again consider the same scenario, right? Same scenario in the sense, say suppose a pathogen has entered inside our body, right? A pathogen has entered inside our body. Once the pathogen enters inside our body, right? Let us take it this way. Say this is a pathogen, right? So this is the pathogen that has entered inside our body, right? Now, once the pathogen enters inside our body, here we have the B cells that are being present, right? So these are the B lymphocytes that are present. On the surface of B lymphocytes, there is a small receptor which is being present. There is a receptor on the B cell. So this receptor is called as B cell receptor, or simply we can call it as BCR, right? Or B cell receptor. On the surface of the B cell, there is a receptor being present. What do we mean by a receptor? Receptor is nothing but a receiving molecules, right? Receptor means receiving molecules, right? Now, this B cell receptor, which is present on the surface of the B cell, we also call it as membrane bound receptor. We'll talk more about this in our later sessions, right? For now, remember that on the surface of B cell, there are certain B cell receptors. So this B cell receptor will start identifying the pathogen. So once the pathogen enters into the body, the pathogen will first bind to the B cell receptor. Once the pathogen binds to the B cell receptor, this is a matured B cell. B cell has been formed in the bone marrow and it has been matured in the bone marrow, right? Once it is being matured, they have certain receptors on their surface, what we call it as the B cell receptors, right? Now, this B cell receptors will start receiving or recognizing a particular pathogen that has entered into the body. Right. Once the pathogen binds to or the antigen binds to the B cell receptor, what would happen is this B cell will now undergo the process of activation. Right? The B cell would now undergo the process of activation. As a result of activation of B cell, what is happening is this B cell will now start producing two different types of cells. Right? Activated B cell upon the entry of pathogen, after it has been recognized by the B cell receptor, the matured B cell will start producing, or it starts activation, or it undergoes differentiation to produce two different types of cells. Right? One would be termed as the memory cells. One type of cell would be the memory cells. The second type of cell is called the plasma cells. Right. So, getting it? once there is some sort of entry of microorganism into the body or any pathogen into the body, the B cell receptor will first identify and recognize that pathogen. Once the B cell receptor recognizes the pathogen, this B cell, matured B cell, will now start undergoing the process of activation. After activation, it would undergo the process of differentiation, producing two different types of cells. One is called the memory cell, and the other one is called the plasma cells. Plasma cell, it is also called as effector cells. So there is another name given for it. It is also called as effector cells or plasma cells. Right. Now this plasma cell, based on the type of antigen that has entered into the body. Try to get this right. Right. So this plasma cell or the effector cell, based on the type of antigen or pathogen that has entered into the body, they would now start synthesizing something called as antibodies. Right? So they would start synthesizing the antibodies. Getting it? Right? So what we are seeing is, we are talking about what is the role of the B cells present in our body. Once there is some sort of pathogen or antigen entering into our body, I hope all remember what antigen is. I told you in the previous session, right? Antigen is nothing but any foreign particle that enters into a body. Any foreign substance or particle that does not belong to our body, we usually call it as antigen. Right? So be clear with that. So whenever there is any antigen or pathogen entering into our body, the B cell receptors will start recognizing it and it would bind to that particular antigen or pathogen. Once the antigen has bound to the B cell receptor, this B cell would now start undergoing the process of activation, and the matured B cell would get differentiated into two types of cells. One is called the memory cell and the other one is called the plasma cell. The plasma cell, based on the type of antigen that has entered into the body, the plasma cell will now start producing specific antibodies against the specific antigen that has entered into the body. Now, this antibody will go and start targeting this pathogen, and it would bring about the destruction of the pathogen that has entered into the body. That's how the B cells usually work. Clear? Right? So how does the antigen destroy the antibody? Sorry, how does the antibody destroy the antigen? That will take up little later once we have done with all these cells, everything. Right? So how does the antibody bring about the destruction of antigen? That we'll see little later. Right? So there are different ways how the antibody will target the antigen that has entered into the body and it would bring about its destruction. So all that we'll see little later. For now, just understand the role of B lymphocytes in our body. Right? So what exactly is happening? One more time, just briefly one more time. So what exactly is happening? Whenever a pathogen or antigen enters into the body, at that point of time, the B cell receptors present on the B cells, matured B cells, will start recognizing it. Right? It would start recognizing the pathogen or antigen. Once it recognizes, it would bind to the antigen or pathogen. Once it binds to the antigen or pathogen, this B cell would now undergo the process of activation. Once B cells have been activated, this B cells would now undergo differentiation to produce two types of cells. One is called the memory cells and the second one is the plasma cells. The plasma cells will now start synthesizing the antibodies based on the type of antigen that has entered into the body. So once the antibodies are synthesized, these antibodies will now start acting on this particular antigen that has entered into the body. Right? And it would bring about the complete destruction of the antigens that have entered into the body. That is the role of antibody. One very important thing what you need to remember, antibodies are formed from the plasma cells or the effector cells only upon the differentiation of the matured B cells. Right? So we know that B cells produce antibodies, but what exactly is happening? B cells would undergo differentiation to produce plasma cells, and the plasma cells or the effector cells are the ones which are actually producing the antibodies inside our body. Those antibodies will go and start targeting the antigen. It will bring about complete destruction of antigen. Clear this? Right. Now, we are left out with one more cell, which is the memory cell. Right? So one type of differentiated cell, that is plasma cell, would produce antibodies and destroy the antigen. Second type of cell, we have the memory cells. Now we saw that yesterday in the previous session, right? We saw that one of the basic characteristic feature or unique characteristic feature of the acquired immune system is memory, their ability to remember the pathogen that has entered into the body. Now start recollecting the secondary immune response what we saw. Right? So in secondary immune response, what did we say? Or rather primary immune response? This first process is all nothing but primary immune response. Right? Entry of antigen, recognition, B cells undergoing differentiation, memory cells, plasma cells. Now plasma cells, based on the type of antigen, it would start producing specific antibodies, and these antibodies would go and destroy the antigen. So all this process what we have, the second bottom one what we have, would refer to as the primary immune response which is produced by the body. We said that primary immune response takes a longer time. Why does it take longer time? Recognition, B cell undergoing activation. After activation, B cells should undergo differentiation. After differentiation, production of memory and plasma cells. The plasma cells should now synthesize the antibodies based on the specific antigen that has entered into the body. That's the reason why it is usually taking a longer time. Whereas, second time if the same antigen enters into our body, right? Second time when the same antigen enters into our body, this process is common. Recognition is common. But this time there is no need of activation of B cell. There is no need of differentiation into plasma cells and memory cells. There is no need of synthesizing the antibodies. But how will it fight again? These memory cells will directly start targeting this particular antigen during the second response, right? So this we started calling it as secondary response, right? And this would be the primary immune response in our body. Right. So we said that secondary immune response is much faster. Why it is much faster? There is no activation, there is no differentiation, there is no synthesis. Directly the memory cells will start targeting the antigen, and the memory cells itself will bring about the destruction of the antigen, because it remembers the antigen that has entered inside the body. This is how the B lymphocytes usually work. Yeah, right. So this is the first type of cells under the acquired immune cells. So this is one of the major constituents of the third line of defense, which is the B lymphocytes. Right. So constituents of the cells, constituents of the acquired immune system, it is mostly constituted by the lymphocytes. Two types of lymphocytes, B lymphocytes and T lymphocytes, and this is how the B lymphocytes usually bring about their function. Right. So B lymphocytes produced in the bone marrow, right, matured in the bone marrow, right? So they are formed from the hematopoietic stem cells present in the bone marrow, produced in the bone marrow, matured in the bone marrow, and they start producing this. Right. Now these antibodies or B cells which are being produced, they bring about the complete destruction of antibodies. With the help of antibodies, they completely bring about the destruction of the antigen that has entered inside the body. Yeah. So this is what we have with B lymphocytes. Right. I hope everything is clear with regards to B lymphocytes. The first type of cell under cells of the acquired immune system. Right. Now let's take up with the second type of cell. Right. The second type of cells under the acquired immune system. The second type of cell what we'll be taking up is called as the T lymphocytes. Right, it is called as T lymphocytes. So let us take up quickly a few things with regards to the T lymphocytes. So the second category here, or the second type of cell, would be the T lymphocytes, or simply we can call it as the T cells, or we can call them as the T lymphocytes as well. Right. Now what about these T lymphocytes? Again, the T lymphocytes are produced in the bone marrow. Again, from what they are produced? They are again produced from the hematopoietic stem cells that is present in the red bone marrow. Right? So T lymphocytes are produced in the bone marrow. Once they are produced in the bone marrow, they have to undergo the process of maturation. So these T lymphocytes, they are not matured in the bone marrow. From the bone marrow, they migrate to one of the organs that is present in our body, which is called as thymus. Right? So they migrate to thymus for their maturation. Right? Things what you need to remember: B lymphocytes, they are produced in the bone marrow and they are matured in the bone marrow. Whereas T lymphocytes, they are produced in the bone marrow and they are matured in the thymus. I hope all of you remember this organ called thymus. We spoke about a lot of this organ when we were talking about our last year, when we were talking about the topic called as the endocrine system. Right? Under endocrinology, we spoke about this organ called as thymus, a small organ which is located very close to the heart, whose major function is to secrete a hormone called as thymosin. Right. So there we said, right, the role of thymosin is to bring about the process of maturation of the T lymphocytes. Right. So thymosin is very, very essential for the process of maturation of one of the most important immune cells that is present in our body, which is called the T lymphocytes. Right. So if T lymphocyte maturation doesn't happen, what would happen? Our immune system would actually become weaker. Right? Since it is matured in the thymus, it is being termed as T lymphocytes. B lymphocytes, B word comes from the bursa of Fabricius in case of birds, whereas T lymphocytes, T comes from the organ called thymus, where it undergoes a process of maturation. Right. I hope all would have been familiar about this. Right. So when we talk about the immunity of an individual, right, during the different stages of the human development, right, during the different stages of human life cycle, right, the immunity keeps varying. In case of small children, the immunity is said to be very less. Right. That's the reason why most of the small children are more prone to a lot of infections. Whereas if we move to the adult stage, right, teenage or the adult stage, at that point of time, the immunity is said to be the strongest, right? So the immune system is said to be very, very strong during the teenage time or during the adulthood time. But again, as the age starts increasing, during the older age, again, the immunity will start reducing. Right, the immunity would start reducing. Have you anytime analyzed what could be the reason for this? The major reason for this, what the scientists say, is because of this particular organ called as thymus. Right, we spoke about this in our last year, right, when we were talking about the endocrine system, especially talking about this organ called thymus. Right, I told you that during the childhood time, thymus is actually very, very small. It slowly keeps growing. Right, it slowly keeps growing, and maximum size would be reached only during puberty. Right, maximum size is reached only during puberty. That's the reason why we say the most immunologically strong people are the individuals who are present during that puberty time. That is middle age, right? Middle age individuals, right? Teenage as well as middle age. That is the time where the immune system is said to be very strong. Why? Because the thymus is fully developed in those particular individuals. Again, as the age of the person keeps increasing, the thymus will slowly start undergoing degeneration. We had given it a term also. We started calling it as atrophy. Right? Slowly, the thymus will start degenerating after puberty. As and when the thymus starts degenerating, what would happen in the older people? The immune system will be reduced. Why again that is happening? Major reason is maturation. In children, because the thymus is not completely formed, thymosin secretion is said to be less. If less thymosin is being secreted, the maturation of T lymphocytes is also less. Since the maturation of T lymphocytes is less, the immune system is also said to be weaker. Same applies even in the adult, even the old age also. In the old age individuals, I told you that the thymus will slowly start degenerating. As the thymus slowly starts degenerating, what would happen? Again, the levels of thymosin or the amount of or the concentration of thymosin being secreted by the thymus will go down. As the thymosin level starts going down, again, what would happen? Maturation of T lymphocytes also will start decreasing. As a result of which, these two groups, age groups, that is children as well as older people, they are said to be having less immune power. The reason for that is this one. Right. Okay, getting back to our topic. T lymphocytes are produced in the bone marrow and they are matured in the thymus. Right. Again, from where they are formed, they are also formed from the hematopoietic stem cells from the red bone marrow. And for the process of maturation, they move to the thymus. Right. Now, we have two types of T lymphocytes, right? Two different types of T lymphocytes have been identified. One is called T helper cells. Simply we can call it as Th cells. H stands for helper. T helper cells or simply Th cells. The second one, so this is called as T helper cells. The second one is called TC cells, right? TC cells, right? So C stands for T cytotoxic cells, right? So T helper cells as well as T cytotoxic cells. So there are two subpopulations under the T lymphocytes, T helper cells as well as T cytotoxic cells. Let us quickly take up with what is the role of T helper cells as well as T cytotoxic cells and how do they bring about immune response in our body. First, let us take up with the T helper cells. So talking about TH cells, what does these TH cells do in our body? Right. Again, let us take up the same scenario again. There is the entry of microorganism or say any pathogen into our body, right? So there is an entry of a pathogen. There is entry of a pathogen inside our body. So say the same pathogen here, right? So this is the pathogen that has entered into the body. Right. Now this pathogen, once it enters into the body, we have a specific cell here called as the TC cells. Right? Not TC, we are talking about T helper cells. So we have the TH cells that are being present. Right. Now these TH cells have two types of receptors on their surface. Right. One is T cell receptor, we simply call it as TCR, and the other one is called as CD4 receptor. Right? So there are two receptors present. One is called T cell receptor and the other one is called CD4 receptor. CD usually stands for cluster of differentiation. Right? It is called as cluster of differentiation. CD stands for cluster of differentiation 4 more. So in different types of T cells, right? So we have different types of CD molecules which are being present, cluster of differentiation. We'll talk more about this in detail in our later topics when we have some time and we get into the process of differentiation or the process of activation of T cells. So once we have some time, we'll get into the details of that. But for now, remember that there are two receptors present. One is called T cell receptor and the other one is called CD4 receptors, which are present on the surface of T helper cells. Right. As and when the pathogen enters into the body, the pathogen would bind to the CD4 receptor. But again, there is few more processes before this, right? So don't get confused. So that I'm just going through basic things, right? There is actually a lot of process in between, right? For now, just for your understanding, I'm just putting it this way, right? So once the pathogen enters into the body, it will bind to CD4 receptor. This CD4 receptor will now start activating the T helper cell. Once the T helper cell gets activated, T helper cell would start showing its function in two different ways. Right? T helper cells functions in two ways. One is it goes to the B cells and it would start activating the B cells. That is one way how it works. So T helper cell would bring about the activation of the B cell. We know what happens once the B cell gets activated. Once B cell gets activated, this B cell would now start producing two types of cells. And it would start producing two types of cells. One is the memory cells and the other one is the plasma cells. And we know that these plasma cells would now start producing antibodies, and these antibodies will now go and fight against that particular pathogen that has entered inside the body. This is one way how the T helper cell would work. The reason why we are calling it as helper, because it is helping the other cells to recognize the pathogen and fight against that particular pathogen. This is one way how it acts. Second way how it acts is it brings about the activation of TC cells. Right? It would bring about the activation of TC cells. Right? So once TC cells gets activated, I think we can complete it here itself. So we have done with TH cells. So the role of TH cells is once the pathogen enters inside the body, right? So this T helper cell will first recognize it. Once the T helper cell recognizes the pathogen, it would function in two ways. One is it brings about the activation of B cells. This activated B cell will now start producing memory cells and plasma cells. Plasma cells will now start producing antibodies. Those antibodies would now go and start targeting this pathogen that has entered inside the body. That is how one way it works. Second way how the T helper cells works is it brings about the activation of the TC cells, the second type of cell, that is the T cytotoxic cell. Now let's take up what does the T cytotoxic cell do. From the name itself, we can make out cytotoxic. Cyto means what? Cell. Toxic is toxic. So it is said to be toxic to the cell, foreign cell that has entered into the body. Therefore, we start calling it as T cytotoxic cells. Now, what does the T cytotoxic cell do? These T cytotoxic cell, they have a receptor on their surface, and this is CD8 receptor. This is important things what you need to remember. CD4 receptor present on the surface of T helper cell. CD8 receptor present on the surface of TC cells. Right? It is actually a very lengthy process, just making it simple so that you can understand how they are actually functioning. Right? Now, what does the TC cells do? Upon activation by the T helper cell, the TC cells will now start producing two types of substances. It would start producing two types of substances. One is called as porphyrin, right? One is called porphyrin. The other one is called granozymes or granzymes. Granzymes and porphyrins. Upon activation of TC cells, the TC cells would start producing two types of chemical substances. One is called porphyrins and the other one is called granzymes. Right. Now, what does this porphyrins do? From the name itself, we can make out porphyrin. That is nothing but something related to pores. So this porphyrin, what it does is, it would start making pores on the antigen or pathogen that has entered inside the body. Right? So just imagine that this is the antigen that has entered inside the body. Let us put it this way itself. Right? So this is the antigen that has entered inside the body. Right? So what does this porphyrin do? It starts making pores on the surface of this antigen. So it has now started making pores on the surface. Once the pores have been made with the help of this porphyrin, this granzymes or granozymes will start entering through the pores, and it would start destroying the antigen that has entered into the body. That's how the T cytotoxic cells usually work. Right? So what does the T cytotoxic cells do? Upon the activation, T cytotoxic cells will now start producing two types of chemical substances. One is called porphyrin and the other one is called granozymes. Porphyrins will start making pores on the surface of the antigen, while the granozymes, whatever are there, which have been released, they would directly enter through the pores into the antigen, and it would lead to the complete destruction of that particular antigen. That's how the T cytotoxic cells usually work. Right? So T cytotoxic cells usually give us protection, or it would give us resistance against most of the tumor cells that has been present in our body, as well as several viral infected cells. So whenever there is severe viral infection present in our body, as well as whenever there is some sort of tumors present in our body, cancerous cells present in our body, majorly it is the T cytotoxic cells which actually work in providing resistance to our body. Right? So these are the T cells as well as the B cells what we have, which are a part of the acquired immune system. So quickly going through T cells once again. Right? So T cells are produced in the bone marrow from the red blood cells. Right? That is more precisely speaking the red bone marrow, not the red blood cells, sorry, red bone marrow. Right? And they move on into the organ called thymus for their maturation. right? So once they are matured, they will get divided into two subpopulation, T helper cells and T cytotoxic cells, right? So T helper cells, what do they do? From the name itself, we can make out they help in the activation of B cell as well as the activation of T cells, that is TC cells, right? So they start activating B cells so that B cells will produce memory cells and plasma cells. Plasma cells will produce antibodies, and those antibodies will go and start fighting against the pathogen that has entered inside the body. Whereas on the other side, it would start activating the TC cells. This T cytotoxic cells would produce two different types of chemical substances. One is called porphyrin and the other one is called granzymes. Porphyrins will start making pores on the surface of the antigen, whereas granzymes will start entering inside through those pores, and it would bring about the destruction of that particular antigen that has entered inside the body. Right? T helper cells has CD4 receptors on the surface. T cytotoxic cells, they have CD8 receptors present on their surface. Right? So this is what we have with the cells of the cells of the acquired immune system. Right? I hope all are clear with the cells. Two major types of cells: B cells, B lymphocytes, as well as the T cells, the T lymphocytes. Right? So with this, we are done with the cells of the innate immune system. So in the next class, we'll take up with the structure of the antibody. So we'll get into some of the details of the antibodies. So we'll take up with the structure of antibodies, and then we'll try taking up with different types of acquired immune system that we have in our body. Right?